Thank you very much, Adam. Vice Chancellor, ladies and gentlemen, colleagues. Somehow the system already decided that Adam was talking too long. I don't believe he was. And started the lecture without me, but now I make a proper start. Thank you very much for coming. And thanks to my group for coming. Fantastic, thank you guys. I appreciate the support. So today, what I thought I'd do, you heard Adam listing the interests I have, and it is a quite varied field of different interests. And so rather than boring you with a list of like short bits of all of the things I do, I thought I'd focus on two quite interesting things I'll be doing at the moment and tell you about two major research projects that we are involved in at this time. And I've summarized my talk and the intention of what I was trying to tell you today in these two videos up here. They unfortunately have started to freeze up. But you can see there on the two sides, we had a video of free-flying bees that, as you would appreciate, fly quite elegantly, and they land, and their robust system doing some actually quite sophisticated cognition. And on the right-hand side, I took the outtakes from the DARPA Challenge 2016. These are the robots that failed the most miserably in all the tasks. They kind of put them together, they put all the scenes together and publish it on the web. But the point I was trying to make is, and I think it's still a valid point, even though I'm being a bit cheeky here, is that our technological systems, autonomous robots, aren't quite yet at the level even of simple insects. So simple insects have still something to teach us how to build better robots and better AI. And in the subsequent talk, I want to show you two examples of the kind of things that we're trying to learn from insects. So I'm going to start with, they're working again, look at that. Part one, I'm going to look at how insects do what I call recognizing odor objects, or odor objects recognition. And that is a problem that will be defined while I go along, so bear with me for a moment if that doesn't speak to you at this particular point in time. So to start with, I'm going to introduce a little bit olfaction and the olfactory system of insects, and then I'll come to this particular problem that we are working on. So just to start from the very beginning, olfaction is the sense of smell. So that's what we do when we detect chemicals in the air, and we as humans do it with our nose, as everyone knows. What actually does it is the olfactory epithelium at the top of the nose, and I'll show a picture in a moment just to fill in a bit of background there. Insects do not have noses. Insects have antennae that have the same sensory function as our noses. Now, one of the reasons why olfaction and this odor object recognition question is particularly interesting is because olfaction is a bit more complicated and therefore more interesting to me than vision and audition. And that's because there's some things in olfaction that aren't as clear as there would be if it was about vision. And that involves simple things that we take for granted normally in the other senses, like in vision, if we see something and things are close to each other in the world, then they're close to each other on our retina, and we can map that in the brain. And as long as you keep a retinotopic mapping, a mapping of where things are on the retina, you know where they are in the world. And to some extent, that's also true for the sound. Yeah, so here sound sources are also arranged in the world, and they would be if they come from left and right, you can distinguish that just by coming to the different ears. And in a similar vein, if we think about what we see, colors are similar if their wavelengths are similar, very similar concept, simple concept. Tones are similar if their frequencies are close. Yeah, so that's quite straightforward. And I say all this, I mean, it seems very obvious, but I say all this because it's not true in olfaction. Yeah, we come to that in a moment. But just to give the example of the colors, so here's the spectrum of visible light, and here's the wavelength, and certain wavelengths have certain colors, and you notice it's a continuum. It's not just kind of a checkerboard of different colors here. So if you have something yellow, it's around here, and different yellows are next to each other, and different reds, and you have a kind of proper continuum of colors. And so you know a little bit, it's a bit easier to understand what the visual system might want to do if it sees colors. If it's mixtures of colors, it's a bit more difficult. You get these kind of color gamuts, where the pure colors are on the outside, and you can then have mixtures of them. But it's still something that we can visualize, and we kind of know what the brain would want to reflect and respond to. And the contrast is this. Contrast is chemicals in the air, that makes odors, that is olfaction. And for chemicals, there are certain ways where we could arrange them as similar. So we could say chemicals are similar if they have a similar length of carbon atoms in the carbon atom chain. And that's something chemists do, and then they give the names accordingly. So the alkanes are the ones that have a carbon chain and no other functional groups, and then the alcohols, acids, and so on, and they have different lengths, and then they get names accordingly. So you would have methanol, ethanol, and so on. But the other direction, of course, here's all the functional groups, but then you can also go by spectra of resonance of the molecules. You can go by molecular weight, which doesn't necessarily map to the number of molecules because the atoms are different weight and so on. You have many different ways of how you can arrange chemical space, and it's absolutely not clear what an animal would want to do. I mean, what is a similar odor is a completely open question, and it still remains open today. And the research is active in this area, and still people cannot really answer the question. One way of asking the question experimentally is to ask humans, here are some odors, how similar are they? And people have done that to a great extent, and they have found very interesting things. And I want to show you 2 examples only. They have found that the way we perceive odors as similar or not similar is not intuitive and pretty much doesn't map on these other dimensions I just suggested. There is a bit of a similarity if it's alcohols and they have close chain length, they smell somewhat close, but it's not a guarantee if chemicals have the same structure that they smell the same. So if you look at these two here, they are enantiomers of carbon, so I don't know whether there are any chemists in the room. Enantiomers are chemicals that have the same structure in terms of atoms arranged, but they have a different spatial structure and different chirality. And you can see these have the same atoms arranged in the same way, and yet they smell different just because of the structural difference of the molecule. So 2 different smells for pretty much the same molecule. Whereas on the other hand, these odors here are pretty much indistinguishable to humans. They have completely different structures and they all smell the same. So we don't really know what makes different and the same. And so there's a lot of open questions in there, and that makes olfaction interesting. But there is even more than that. Now, if you now move on and say, what's in the natural world, then in the natural world, an odor is never just one chemical. Odors are mixtures of loads and loads of different components. And I always give this example here because it's fun to look at, and we all are addicted to it. Most of us are, maybe some are not. And that's coffee. So if you look at coffee brew, we can easily identify 25 different chemicals in the airspace over a coffee that we can all detect in terms of sufficient concentration, and they make the coffee smell. And people have also investigated this to some length, so you wouldn't think that it's of so much practical interest, but people got really into this, and they looked at what can I take away and it's still a coffee smell. And it turns out not that much. So it's not that you could just take 2 of these, you mix them roughly together and you get coffee smell. It's not like that. You need almost all of these to get a proper nice coffee smell. So odors in the natural world are mixtures of many, many components. And in those we know even less what makes similar and different, because now we have all these different components in there with different chemical structures that somehow interact, and they all have different concentrations as well to be a proper coffee smell. So that makes it more complicated. And I'm going to pile on to that problem. So this is coffee, so we smell it as one odor. Many components, but it's one odor. On the other hand, if you go in the morning to a coffee shop and they also fry the bacon, for a nice English breakfast. Then you can distinguish that. You're getting all the components from coffee and all the components from bacon. Now I haven't researched how many components there are in bacon smell. I'm sure it's more than one. You get all those components together into your nose, and you're not going, aha, there's some coffee bacon on offer here. But you're going, oh, there's some coffee and there's some bacon. Now how do we do that? How do we do that if we have all these components from one source and from the other one is all mixed in the air? How do we even know it's two different things? So there's an extra level of complexity there, and that's where we now approach the question of odor objects. Yeah? So odor objects, in my mind, are the different sources of odors that are meaningful to animals. So in this case, the odor objects would be a coffee and a piece of bacon. And the question is, how do animals disentangle the mess of chemicals in the air to smell what odor objects are in the environment? And this is the question we're trying to address. Now, the last thing I wanted to add is, this is just a side remark, that there's obviously also the problem of different concentrations, and it's known that with chemicals it's harder than with vision, that sometimes we're getting it wrong, and the same chemical can smell different if it's at higher concentration. So that's something that I just wanted to have mentioned for completeness. Now, we're coming to the end of the overview of how complex olfaction is. With this one, and this is a bit of an aberration then, because it adds complexity, but we'll see later that it actually helps us solve the problem. Odors in the environment, unlike I originally thought when I came into this, they don't distribute evenly. They don't distribute in a laminar flow, as they would say in dynamical systems. They are in a turbulent regime. So even in a room like this that has fairly still air, there's no wind in here, yeah? Even in a room like this, odors would distribute like that in a turbulent way, and not in a laminar flow where there would be a gradient of concentration of the odor, where you have the odor source and then there would be odor all around it and the concentration would go kind of gradually down. It's not like that, it's more like this. And if you have two odor sources, and that's depicted here as the red and the blue, in this case it's smoke to make it visible, but it's the same for odors, then they will mix in these kind of irregular ways, and if you were an insect to fly into this, you would get a whiff of this odor and that odor in rapid succession. You wouldn't get the full mix of things all at once. Okay, so that was the bit about what is olfaction, why is it complicated, why is it interesting. Now I just thought I'd give a really, really brief overview of how animals do it in principle. That doesn't answer the question about the complex problems, because this just is kind of the basic anatomy of how we smell. And then I come to our work that looks at how we do the complex bits. So in humans it looks like this. You have a nose and you breathe in some air with chemicals, and then they will here interact with the cilia of olfactory receptor neurons, and that will translate that chemical signal into something electrical, some voltage change, and that's transported in the brain and then further processed up here. In more detail, it looks a bit like this. So this is here the top of the olfactory epithelium that's within the nose, behind the bridge of your nose. And that's where the odorants come. They go into the mucus, into solution, then interact with these cilia, and then you get the transduction into electrical signals, and it's all electrical from there. And you have processing in the brain, first in the olfactory bulb, and then in higher stages like the peripheral cortex and so on. Now in insects, I told you they don't have noses, it looks slightly different, but the olfactory system is actually quite similar. Now slightly different, I think you can appreciate if you compare this is a fly head, here's my head, it's not quite the same, we agree, yeah? And the nose of the fly really is this structure here, this is the antenna. And on there are these sensory hairs or sensilla, and the olfactory receptor neurons of insects sit in these hairs, as opposed to in the top of our nose. But if you then get to the actual receptor neurons, it's a similar method, yeah? So there's also binding of the odorants to the receptor neurons, translation into electrical signals. From there on out, you have these structures here called antennal lobes. They are like the olfactory bulb in humans, it's organized in glomeruli and so on. So from there on out, this is quite similar to what we do. So we can look at the insects and we can actually learn something that's more generally applicable to most animals. Right, now how's the system then working in principle? Yeah, I already said there's transduction of chemicals into electrical signals in the olfactory receptor neurons by a number of olfactory receptor types. Let's say the bee would have 160 different types of receptors, a fly has 50, humans have a few hundred. And it's organized in a way that the different receptors are expressed in the receptor neurons, but each receptor neuron only has one specific type in them. So by a certain neuron being activated, you know which type of receptor was activated by the odor. And then it's further organized in a very sensible way, actually, that all the olfactory receptor neurons of the same type converge onto the same of these little balls, these glomeruli, which means that if you know which glomeruli is on, you know which odor, which olfactory receptor was activated. What makes it a bit complicated again is though that the receptors have a quite broad profile, which means that several chemicals will activate a given receptor. And the opposite is true too, that an individual chemical will activate more than one receptor. So you have this kind of, they call it the combinatorial code, where you give a chemical to the system and then there will be several of these glomeruli lighting up with activity. And it will be overlapping. If you give another chemical, it will be also several different glomeruli, but some the same that light up. So you have this kind of combinatorial code of overlapping patterns. That's what encodes olfactory response. And that's what the brain needs to make sense of if we want to, if the brain should react to odors in a specific way. Okay, right. So now we've got the basics down. I think. I hope everyone's still with me. Now let's talk about recognizing odor objects. So just to recap real quick, odors are complex mixtures of numerous chemical substances, and they mix in these complex plumes in the environment. And the question is, how do we make sense of that? And how specifically can we tell whether there were two sources of coffee bacon, one source of coffee bacon or two sources of coffee and bacon? How do we distinguish that? How do we make sense of that? And our hypothesis in this particular case was that this messy, crazy structure of the plume actually helps, because if you fly in as a bee, you either get green or green or red, but you don't, not very often at least, get green and red. Now, if you were a really fast smeller, then you could actually distinguish that from getting them both together as yellow, so to speak. Yeah, because you get red, green, red, green, red, green, you notice that it's not yellow, it's red, green, red, green, red, green. So you can see it's coffee and bacon, it's not coffee bacon. Yeah, so the temporal structure of the plume might actually help to distinguish between a single odor source of well-mixed odors and two sources of not well-mixed odors that just mixed in the air in the plume. Now, is that a reasonable hypothesis? Well, I'll show you that we tested it in the animals, but before I do that, I thought I'd just show you that you can do this kind of distinction. And for a moment I thought I'd bring some coffee and bacon, but that actually wouldn't work. So I thought we'd do something else. I'm going to do a little psychophysiological experiment with you, which is about hearing. In hearing, we have the same problem. It's commonly known as the cocktail party problem. And it's the problem where you have many people in the same room and they're all talking at the same time. But it turns out that when people talk, they don't talk exactly at the same time, but it's more like that plume where someone says something, someone else starts a bit later to something. It's overlapping for sure, but there are differences in timing and onset of voices. And it turns out that our auditory system in our brain is making sense of the cocktail party mix of voices with the same mechanism of the question of when does the onset of a tone happen, and we will group all tones that happen at the same time together as from one speaker, and all tones that are offset in time we will attribute to a different certain next object. And here's a little demonstration of this effect, and I hope it will work for everyone. So we'll see what I can manage. Figured out that the PA system needs here special treatment for this particular thing to work. All right, here we go. So what I have here for you are two examples of a tone that's put together of pure tones, and this is just like a diagram. It doesn't show anything real other than that there are 500 millisecond long tones, and there are several tones here shown as lines of different frequency, and they're not harmonics of each other, so it will have a bit of a dirty sound. In one case on the left here, all come on together, and in the right case here, one of the frequencies comes in later, and the delay is about 160 milliseconds. And now let's have a look. I'll play them to you, and we'll see whether you can see or rather hear a difference between the two. Okay, and then please do not go up. It sounds a bit like an alarm. It's not an alarm. Right? Don't get up and leave. That would be very sad. So let's have a listen into these. Number one, synchronous, sounds like this. Okay, that was a few repeats of that. Now number two sounds like this, and see whether you hear the difference. Now don't stop. And that's how we feel the rest of the time now. Now can I ask, who heard the difference? Just quick show of hands. Oh, the PA system is better than I thought. All right, that's very good. And your brains, obviously. Who's going to describe to me what the difference was? A volunteer, someone. Two tones in the second one, yeah? Like a blue siren. Yeah, actually it has the second tone, yeah. Okay, Ron?
It sounds like a sweet, like a wah, like a sort of like low frequencies and high frequencies, like wah, wah.
Right, so it's the two-tone thing, right? Low, high. So what happened?
But I also heard the latency somehow.
Yes.
Within that tone, because something was coming on within it.
Yes.
Kind of like
Very good, very good. Yeah. Yes, I like to hear that. So, so here's the thing. No, you get an A, but you need to stay. So here's the thing, it all makes sense in the end, but it's surprising on the other hand as well. So if you just think about the tones at the end here, they are physically the same thing, right? We have all the frequencies here, all the frequencies there. However, as you reported, like it sounds to us as if this was, the front here was like this tone, and at the end here it's like this tone plus a high frequency tone, like this E kind of, da-dee, da-dee kind of tone. So what's our brain doing to us? Yeah? Basically, the brain says, oh, look, this tone came too late. It's not part of this overall tone. It's a separate one. It's a second speaker. Yeah? It's a second person. And therefore it's separated out. And even though the physical tone here at the end is the same as this one, it sounds to us here in the beginning as the same as that because we don't notice the missing thing. There's too many others. And the second new one in here is a new tone. And the brain keeps that information for the whole duration of these 500 milliseconds. There's no, these are two. It's not one. There's here, it's one. And you can see how that kind of corroborates my story, right? So whenever things come together, they're one tone. And if they don't come together, there must be a separate tone. Right? So human hearing already does it. Now let's have a look whether insects smell that way. And we see whether we can go there. Now, the problem with insects is, I'd rather like to do what I just did with you. I would say, hey, here, bee, here's some smell. Did you smell the difference? And they would go, ha, well, of course I did. Or maybe, no, you didn't. But unfortunately, as you might know, they don't talk. So what do you do? Well, people smarter than me have invented a way around that. There are other ways to ask insects what they perceived and what they smelled. And that works like this. This was invented by Randolph Menzel, one of the really big names in insect olfaction, a professor in Berlin. He's now retired, but he's still very active. He invented this idea of the conditioned proboscis extension response in bees, which is an experiment where you do the following thing. You take the bees and you give them a signal, let's say an odor, and you pair the odor with sugar water. And the bees go, aha, this odor, yummy sugar, and they start licking it, first of all, but also if you give the odor again afterwards, they will try to lick. no matter whether there's sugar or not. They think this odor means sugar, let's go. Yeah, and you can see this is a little movie here. If I can get this started. No, that wasn't it. No, mouse. Here we go. Going back. Right, here we go. This is a trained bee, so that bee has experienced the odor and sugar water before. And when this little square comes on, the odor comes on now, and you see there's the proboscis, yummy sugar. Even though there's nothing there, right? So the bee has learned this odor means sugar. And that we can now exploit quite smartly. So what do? Coming back to the odor objects. We train the bees to respond to odor A. And I just call it A for the moment. This is some odor. Yeah? And then we test them with these different mixtures of that odor A with an odor B. And what's the difference between the mixtures? The mixture is either that is fully mixed and they come on together, or it's odor A and then B, or it's B and then A. And we give a little delay. Yeah? In this particular example, 6 milliseconds. It's a tiny delay, actually. This is to scale. This is 800 millisecond long bar. Then 6 milliseconds would be like this offset here. Who can see the offset? Oh, good eyes. All right. Yeah, but it's not big. I hope you agree. Okay. Now, the question then is, do the bees smell A in that, and do they smell it different here than there? And we know if they do or not because they will either start to lick or they won't start to lick. Right? So we can see what they smell. And here are the results from that experiment. And you can see this is the training, the number of trials where they got the sugar water, and they go from not extending the proboscis, like 10 of them do it randomly anyway, 10%, but it goes up to like 60 plus percent reliably extending the proboscis. This is normal, so bees have this thing, they either learn or they don't. This is not that they sometimes extend or sometimes don't. It's just 40% they learn absolutely nothing. And then the experiments are done in this way. You just train them all, then you take the 60% that actually learn something, you go on with them. So that's what happened here. and then the different test conditions were given. And you see 50% of the ones that had learned something. So if you had just given A, there would be 100%. Now if you give A and B together synchronously, you get 50% responding. But if you give the little delays, A and then B, B then A, you get a significantly higher number of bees responding. So that means the bees do smell A more when it's asynchronous. And that's like with the tone, right? When it was asynchronous, we heard the extra tone. And if it's synchronous, we don't. If it's synchronous, the bees are a bit confused. Some do think A is in there, some don't. But if it's with a delay, they're much surer that there is odor A in there. So this is a work of my collaborator, Paul Suska, and a student, Georg Stierle. And that's where I came in, really, where we said, now, that's great, so they can do it. How do they do it? Do they have any hypothesis, any kind of model, how we could explain how they do it? So I went ahead and we built a model. And the way you build models in computational neuroscience, I appreciate not everyone's doing computational neuroscience, the way you build them, you need to combine what you know with what you think you want to test. So there's two elements to a model, always. There's a data-driven part where you really just assimilate everything you know about the system. And then there's a hypothesis-driven part where you think, I think the system works like this. And then you try to implement that into your model within the constraints of what actually is there. I should say, this may be not understood. If you just thought you could just build it fully data-driven, there's always a bit missing. There's just things that you don't know about the system, so you cannot put it in your model. It might be the strength of these connections between receptor neurons and projection neurons in these glomeruli. It might be how many are connected to how many. There's always something missing. You don't have a full picture of biology. Maybe at that point we would be done. Arguably, we can argue about that later on with a glass of wine. But that's not even the case. We have always loads of holes in what we know, and that's we fill in by putting a hypothesis, and then in the computation model you can test whether that all makes any kind of sense or not. So you implement it, you simulate it, and then you can see whether what you thought might happen actually happens. So what did we think might happen here? This is now where it's getting a bit more complicated, but I hope I can explain that. what we thought might happen here is that what we need to achieve is that when it's a synchronous mixture of two things, that looks different than if it's asynchronous. And that could be achieved by something, a circuit like this, where you have these local neurons, and they're known to exist, and they inhibit each other, which means when one is on, the other one can't be on. Okay? Now, every odor would have a preferred OR type that they activate most. Yeah, just by the fact that there's a finite number of them, you can find the one that is activated most. And typically, for two different odors, it would be two different ones. Yeah? And that's just this mini sub-circuit of this whole thing I showed before. There's many more types of glomeruli in here. This is just for two. Yeah? Now, if we then have odor A and odor B, and that happen to be the olfactory receptor neurons that are activated most by B, and these are activated most by A, then whenever I put in odor A, then these would go on first because they're most activated, so it's much stronger response, faster response. They would switch on their local neuron, and it would switch off the other one. And even if the odor B comes right after, this one switched off for good, so it can't respond. And the information, which one was first, is saved in the fact which one of these is on. Because if B is first, this one goes on, that one goes off, and it stays that way, no matter whether I activate this really strong with A afterwards. You see the logic is like a little memory switch, like a little memory circuit there. And if that works, that could then modify the projection neuron pattern in a way that makes these two different conditions different, and potentially different from when it's a synchronous mixture, which might have yet a different local neuron that is switched on. Or it could be one of those. That's something that's not under my control. Okay, so that was the hypothesis, and then we went in and we tried to test it, and this is just a very quick couple of slides of data before I come back to somewhat lighter material again. So here we can show that in the local neurons this works. So in the full model we have all kinds of different glomeruli. That's the different matches to the different olfactory receptor neuron types. That's these numbers here, and turns out for these particular odors, these are actually particular odors that we are trying to simulate. This is hexanol and nonanol. It turns out that glomerulus number 28 is the one that's on for hexanol. And the one that's on for nonanol is the glomerulus 33, yeah, the local neuron of that glomerulus. Yeah. And you see it works if you then do a mixture of hexanol, 6 millisecond delay, nonanol, then it's the hexanol one that's on, 28, and not 33. If you nonanol first, then hexanol, then it's the nonanol one on, and not the hexanol. Okay, so this kind of little memory circuit works. That wasn't guaranteed, I should say, because of course there's noise in the system, and the synapses aren't infinitely fast, so it could have well been that there's loads of mistakes, where by mistake the wrong one gets on. Isn't the case. Yeah, that seems to be within realistic limits, that seems to work. And then you get the projection neuron pattern, and it's modified by these local neurons that are on or off. And as you can easily see, that explains everything. Now, as you can see, you can actually not really decipher this very well, because it's this combinatorial code I was talking about. Several glomeruli are on for all these different conditions that I'm plotting here. And yes, one can see that magenta is a bit different from cyan. Yeah, that's the two delayed conditions with the different odor first. You can see there's differences, but how different, not what kind of sense it makes, is really hard to see in the raw data. But that's why you do data analysis. So in this particular case, last data slide here, we did just the correlation analysis where we said, oh, we're going to define the prototype response to A, prototype response to B, what are those patterns? And then I take the correlations of the mixture responses with those prototype responses to tell how they look like. If there's large correlation, I say it's A. If it's a small correlation, it's not A. Yeah? So just that simple kind of idea. Here on the left, I'm not going to show that. This just shows that the method works. Yeah, I just compare here like hexanol to hexanol template, nonanol to nonanol template. That should be 100% correlation. Sure enough, they're up here at 1. Yeah, so the method works. And then I look at the interesting bit, which is the mixture conditions. Now I have here two different types of delays. Maybe we concentrate on the small one. That's again the 6 millisecond delay, the really small one. And you can see here that if I now compare the mixture of hexanol first, then nonanol to nonanol, the nonanol template response, then I have a fairly high correlation. So hexanol first, then nonanol looks like nonanol. And the synchronous mixture looks not as much as nonanol. That's this color here. Yeah? It's less. If it's the other way around, if I compare hexanol then nonanol to hexanol, it doesn't actually look so much like hexanol. So that's a bit surprising. This is actually only explaining half the story. It actually explains that you can see the second odor in the delayed mixture very well, better than in the synchronous mixture. That's congruent with what we saw in behavior. That's right, so to speak. Yeah? This other bit's actually a bit wrong. Yeah? So the other way around should also have been higher than the synchronous mixture. So we went through and did this with all kinds of odors and all kinds of glomeruli to look at, and we found that sometimes it fully works, sometimes it's mixed like this, yeah, sometimes it doesn't work at all, which tells us that this model has a bit of a, I mean, it captures half of the story, but not the full story. And that's why this whole thing still continues. Yeah? So we haven't the full answer yet. This is a possible explanation for half of what we see, but not for all of it. And we're continuing this. This is a Human Frontiers program. So as Human Frontiers is set up, it needs to be intercontinental. International isn't good enough for them. Okay, so we have groups from Germany, Japan, the United States, and here at Sussex. And we're still working further on these questions. And some of the guys who are working on the project are in the last row. Right. Now, that was the bit about the odor objects, odor object recognition. And I can see that I'm already running slightly late, but I wanted to tell a second story about what we call the Brains-onboard project, where we're looking more at the robots, because you remember there was bees and robots on the first slide. Now what about the robots? And I thought as an introduction, I just show you a little bit what the competition is doing, and then I show you a tiny bit of what we want to do, because we haven't gone very far with this yet. So the classical approach to controlling a flying robot is control theory. And the idea behind that is that you make a model, a computational model of your drone and what you want to do, and then you do control theory to know how you need to steer it based on very good data of what it's doing at the moment. So it's very well explained in this TED talk of Raffaello D'Andrea, and I just cheekily took two clips from his TED talk to explain you the highlights of that kind of approach. So here's the first one.
Quads are extremely agile, but agility comes at a cost. They are inherently unstable, and they need some form of automatic feedback control in order to be able to fly. So how did it just do that? Cameras on the ceiling and a laptop serve as an indoor global positioning system. It's used to locate objects in the space that have these reflective markers on them. This data is then sent to another laptop that is running estimation and control algorithms, which in turn sends commands to the quad, which is also running estimation and control algorithms. The bulk of our research is algorithms. It's the magic that brings these machines to life.
Okay, so points to note. I mean, I'm going somewhere with this, so I'm going to make a commentary on it. It's not about that this isn't really exciting research, so don't get me wrong, but it's slightly different from what we want to do. And one of the differences is that you already heard here is that this person, he depends strongly on having a global positioning system, knowing at all times exactly what the quad is doing, and he's depending on a camera system in his room. We have one of those as well, actually, over there in Chichester. But he's depending on that to make the decisions. So whenever he's outside the room, the drone would be completely helpless. Right? It's just depending on this global positioning system. And then it's control theory from there to calculate what the drone is supposed to do. Okay. Now the next bit is actually very impressive. So let me show you this one.
You can notice that this quad is making fine adjustments to keep the pole balanced. How did we design the algorithms to do this? We added the mathematical model of the pole to that of the quad. Once we have a model of the combined quad-pole system, we can use control theory to create algorithms for controlling it. Here you see that it's stable, and even if I give it little nudges, it goes back to the nice balanced position. We can also augment the model to include where we want the quad to be in space. Using this pointer made out of reflective markers, I can point to where I want the quad to be in space, a fixed distance away from me.
Right, so there you go, and very impressive, yeah. So they can balance this pole, which is sure not an easy task. Even drive the drone around while it's balancing the pole. It's not dropping it. It's not glued on there, right? There's really good control in there to keep this pole upright. Well, then again, you heard the explanation. They added the pole to the model of the drone. That means whenever the pole is a bit shorter, longer, heavier, different, they would have to start over from the beginning, because they would have to add the new pole to the model of the drone and do all the control theory again. So this is not a parsimonious solution, and this is not how animals work or how we work, where when someone learns to balance a pole, they can balance any pole almost immediately. Yeah? They get a new pole, they're like, how's this one? Oh, okay. Right? And this is not like that. So we're not capturing what animals can do, what bees can do for that matter. And that's more what we are after. A little bit more like what we want to do is this approach here from, again, his colleagues in Switzerland. They're really strong in this area, by the way, so a real hotspot next to Sussex. Right? But they're doing what's called deep learning to have autonomous drones follow paths in the Alps. What's deep learning? Deep learning is basically, I described it here as the artificial neural networks of the 80s on steroids, which is basically just trying to say it's a technology invented in the 80s, depends on calculating, like having big training sets and calculating in a training phase the errors that the neural network is making, and then back-propagating these errors through the system to get a better system. And it's very successful. Here's a couple of examples. There's huge breakthroughs in picture, video, speech recognition. It plays a role in the self-driving cars and all of that. You might have heard about AlphaGo. AlphaGo is the program that plays Go and beat the presumably best Go player in the world, Lee Sedol, just recently. It can also be applied to the autonomous drones, and I wanted to show you this clip of that. To just the last thing of the competition here, yeah? How it follows mountain paths.
In this research, we design a controller to make a quadcopter follow forest trails autonomously. These trails are used by hikers and mountain bikers, and this trail-following technology allows the quadcopter to quickly explore a wilderness area for search and rescue operations. The robot is equipped with a forward-looking color camera, which is its only sensor for navigation. Discerning these trails directly from camera images is an extremely difficult pattern recognition problem, which is also challenging for humans. In the images you see here, it is not at all trivial to determine which direction the trail goes. To solve the navigation problem, we designed an image classifier that takes an image as input and returns one of three classes indicating whether a trail is visible on the left side of the image, in the center of the image, or on the right side of the image. When the robot is on the trail, our controller applies the classifier to the image acquired by its camera and steers the robot in the corresponding direction in order to remain on the trail. The classifier is implemented using a state-of-the-art deep neural network with 10 layers, 150,000 weights, half a million neurons, and 57 million connections. We allow our system to steer a real quadcopter. 10 times per second. Controller acquires an image from the front camera and processes it with our classifier. If the classifier decides that the trail is straight ahead, the robot accelerates. If the classifier suggests that with high probability the trail is heading left or right, the robot gently steers in the corresponding direction, decelerating somewhat. In these videos, the robot is completely autonomous, and there is no human intervention. The system was tested both on a parent AR drone 2.0 and on a custom-built quadcopter. For the AR drone, the controller and classifier run on an external laptop connected to the drone via Wi-Fi. The laptop receives the video stream, processes frames, and sends steering commands. In contrast, the custom-built quadcopter is equipped with an onboard odor computer, which runs the entire video processing and control system. In the videos you see, the AR drone navigates a mountain trail without any human guidance, purely using the information obtained from its camera. As you can see, the deep neural network classifier provides excellent information that lets the quadcopter safely navigate a variety of trails that it has never seen before. These results are still in a preliminary stage, but suggest that deep neural networks can be used to provide effective control signals even with high-dimensional and complex inputs like those you have seen here.
So that's the kind of thing we want to do. However, we already know that humans, animals, they're not deep neural networks. And one of the big differences is that to make deep neural network do this, they need to show it millions and millions of images of mountain paths to work this out. Whereas if you have some humans and you send them into the mountains, they can work out what the path is, beyond, having been trained for millions of times. Other examples in them, they have made these deep networks play Atari games, and we can play an Atari game if you explain the rules in 5 seconds. They needed millions of examples to play an Atari game. So it's a different kind of learning. It's very successful. I don't want to take away from it. It's a very exciting direction, but it's not the kind of thing we're after. We are after the things that evolution developed over millions of years, that are kind of parsimonious solutions to problems that then allow us with few examples to learn what to do and how to do it. And in our project, we are trying to kind of re-engineer the bee brain to get these kind of autonomous abilities onto drones. That's the key. And there was the Green Brain project. It was running from 2012 to 2016 with partners in Sheffield, and we started this process. And then now we have the Brains-on-board project where we're continuing on this path. And our approach to making it happen was this year, and this is kind of where I come in more strongly now as opposed to Sheffield, is that in order to do what we're trying to do, you need to simulate a brain on an autonomous device. And to simulate the brain needs a lot of computational power. And there was this twist in the development of computers, maybe unnoticed in the general public, but there was a problem in that computers got always faster and then they suddenly stopped. I kind of took this here as an illustration. You can see here, this is the clock speed of CPUs over time, and you see this is years. So in year 2003, something horrible happened where suddenly the clock speed completely flattened out. And that meant your computer didn't get any faster. You bought a new computer and it wasn't actually running faster. This is something that's known as a law, that basically every 2 years the computers used to be twice faster. And it stopped doing that. And what happened then is that, and this has thermal reasons maybe, they got too hot, you couldn't make them any faster. Okay, but what happened, people of course tried to find different ways of making computers faster, and the different ways of making computers faster is making them more parallel. Just stick in 2 CPUs instead of 1, or 2 cores into a CPU instead of 1. And so everything started to become parallel. And interestingly, that meant that a different technology that was used for graphics processing, these GPUs, they were already far ahead because they had been doing this for years before, because graphics was such a parallel problem. So they had these GPUs that already had loads of cores. So here's an example of a Tesla V100, has about 5,120 cores in it. Whereas the most modern, normal mainstream computers, they're tootling about 12 cores, 16 cores. Yeah? So they're very ahead. And that's the reason why these GPUs are now the core technology for a lot of AI learning and for our project. And it's something I got onto very early because a friend of mine just said, look, there's this new thing, GPUs, let's buy one. And he just put it under my desk and I had to use it. And it ended up to be an exciting direction and really fun. Now I appreciate that we're running a bit late. I have a couple of slides here to explain you why programming a GPU isn't that easy and that it needs some really good thinking. And the main reason is it is not complex, it's only complicated. The problem is that you have loads of cores. So this is an example of the modern V100, and you can't even see it, but each of these little boxes here is a streaming multiprocessor where each of these little boxes is a core. So you can see you have loads of these things, and now you want to solve a problem, so you have to kind of feist your simulation onto all these cores. And a simulation here typically looks like that you're solving some differential equations or some normal algebraic equations, and somehow you need to feist these different equations onto all these cores. And it's a horrible bean counting problem, as I would call it, because somehow you need to match 5,000 something cores to 10,000 neurons, and they just seem to all kind of need to fit on there, and it's all really horrible. And I realized that while trying to do it for a while, I did it for a year or so, and then I said, okay, and this is not working, because then I wanted to change the model with a few neurons more. I had to start over, it didn't fit anymore, and it was just absolutely awful. And I said, no, this isn't working. And I started doing this thing, which up to date we're still using. I call it GPU enhanced neural networks. And what it is basically a meta compiler, a program that writes the program for me. Because now I'm too lazy to do this bean counting, I just wrote a program to make the bean counting. So basically I just input what model I want, then this program is running over here and it makes me code that does what I want. And that's the thing we need, okay? And that's kind of the technology under the Green Brain project and the Brains-on-board project. And at the same time, that was just lucky, NVIDIA decided that they also want to do embedded devices and put them into phones and robots. And they took their massive GPUs and they squeezed them onto these little tiny computers. This is a credit card. So you can imagine it's about the size of a credit card. And this thing has 256 cores on them and goes, you can do some 1,000 gigaflops. And so we can put our simulations now, not through Wi-Fi like in the video in the one computer, we can put them right on board of our drones. And this is a prototype in Sheffield from the partners in Sheffield. And you see the computer is riding right on here. Yeah, that's the computer. That has the whole brain simulation on it, and then it drives the drone around. And how does it look like when it's successful? Here we put a brain model of ocular motor response. That's the response the bees have to not riding into walls. And you can see this robot completely autonomous, without having been trained either. It's in the structure of the model, knows how to avoid walls. Okay, so now I'll stop it here because I think I've had you long enough. I just wanted to end on thanking a few of my collaborators over the years. Now I say a few and have loads of names on there, but it's literally not, it's like a 20% or so of the people I actually work with, but it's the most recent ones, and I'm really very grateful to be able to work with all of these people, very, very smart minds and hard workers. And down there is a few of the funders I'm currently and recently been funded from with. And I thank you very much for your attention. You're a great audience. Thank you.